So, but also a very well-known surgeon performing translational research in surgical oncology. He is a full professor at the Ghent University. He is the director of the experimental surgery lab at the same university, and of course, a senior staff with responsibilities in GI surgery. So thank you for accepting our invitation, Wim, and the floor goes to you now.
Thank you, Olivia, for the kind introduction. I will now share my screen. Is it working?
Not yet.
No, not yet. Yeah, did we jinx it?
What about now?
Yeah.
I think so. It's your screen, not yet your presentation.
Not your presentation yet.
So you see the slide now?
Still just the Zoom.
Yeah, I have it now in presentation mode. It's not in presentation mode?
With your screen.
But not a presentation?
No.
No.
Okay, I'll try again.
Yeah. So if you go to share screen and if you pick straight the slides.
What about now?
Yes, perfect. Thank you.
Okay, sorry for that delay. So I have no disclosures other that I'm a member and a board member of the SO. So to start, what is translational research? In fact, there are some accepted standard definitions, and it's very, very broad. And you see that here. And it's defined as T0, T1, T2, etc., etc. And so T0 and T1 refers to the translation from bench to basic, let's say, in both directions. And then there is a translation of findings from early proof of concept clinical trials to phase 2 and later phase trials. And then there is translation from the trial setting to the broader community. So that's a very broad field and very encompassing. And obviously what is relevant for surgical oncologists is in fact this T0 and T1 part. And it's so mainly bedside to bench research that is based on the unique opportunity that we have as surgeons to have access to samples and to tissue samples, but of course other biospecimens like blood, plasma, stool samples, etc., are very well possible. So I will focus on this type of translational research, which is, as you realize now, just a small part of what is encompassed by this term in general. So the focus of translational research is mainly on biomarker development, on the development of predictive models that predict response to therapy, on prognostic models that predict the outcome of therapy, be it a surgical therapy or a medical therapy, based on the specific molecular assets of the tumor or of other tissues in the patient. So that's the main focus nowadays of translational research in the setting of surgical oncology. But of course, there are many other examples. And there is research that involves devices, instruments, because those are our everyday tools. There is a huge interest nowadays, and you will certainly know that, on the role of the gut microbiome on a range of phenomena, including cancer, but also Alzheimer, infectious diseases, etc., etc. And then imaging is a part of translational research that has taken off considerably in the field of surgery and surgical oncology, but of course in combination with laparoscopy, and enhanced imaging methods and tools are now being explored, and I will give you some examples of that. Now, translational research is a nice example of the fact that research in general, but medical research specifically, is more and more being executed in a multidisciplinary setting. And we know that as each of us gets more and more specialized in a very small domain, it's really more and more difficult to get a helicopter view of what is happening and of cancer treatment in general. And that's why we need to collaborate. And so the best studies and the highest impact studies are now being performed by interdisciplinary and multidisciplinary groups. And this is not a theory, this is actually happening, and this is a graphical result of a kind of nodal network of disciplines that is generated from literature data. And you can see that clinical medicine here, in the left upper corner, is really now connected with a whole range of different other disciplines, engineering, physics, chemistry, computer sciences, AI, etc., etc. So the field is rapidly evolving, but it's evolving in such a way that it is going towards interdisciplinary research. And in fact, as an aspiring surgeon scientist, you need to be aware of these, of the capabilities of these different fields. And so if you want to be successful, find a niche and get involved with these, with the expertise of these other disciplines, of the tools that they use, and follow a course in, let's say, artificial intelligence, programming in Python, etc., etc. So this is a similar graph that shows you across a range of disciplines which one are really very interdisciplinary at the core. And that's the right upper side of the graph. And you see general biology, physiology, and of course has a link to many, many other fields. And then those who are very much stuck in a niche on the left lower side are the nuclear physicists. You can imagine that they are really collaborating with very few people who can understand what they are doing and for whom their work is relevant. But also you see that in yellow there. So the yellow dots, clinical medicine is relatively uninvolved yet with other disciplines. And so it's getting started. You saw that on the previous slide, but there is still quite some room for improved collaboration and interdisciplinarity and with other fields. And we know that this enhances quality if you collaborate with other specialties and other disciplines. And many universities, including our own center, Ghent University, offers in fact grants that are specifically dedicated to interdisciplinary research proposals, where it's really important that you can show that they are on an equal footing and that they have an equal contribution, and that it is not the fact that one discipline is just giving samples or is secondary to the main discipline. No, they have to have demonstrated equal contribution and an equal importance in that type of grant. And I can imagine that many of the countries where, or the centers where you work, have similar types of grants, and that's of course an ideal opportunity to submit grant proposals in the field of translational research. So what do you do to get started with this type of research? Well, first of all, biobank. I will come back to that in a few later slides. And then you need to identify research partners. So, and it's not that difficult. Of course, in physics and in computer science, you need to come up with a very interesting proposal, and you need to spend some time and effort to convince them. But in the field of biology and molecular biology, it's often the case that these labs are really having a hard time to find samples and to find suitable human samples. And so it's an ideal win-win situation. And if you are the partner who has the samples and the clinical expertise and the knowledge and the knowledge how to organize clinical trials, etc., and then they have the equipment, they can do the RNA sequencing, the single cell sequencing, etc., etc. So you should find partners. You should collaborate, and you should be open to collaborations. And then, of course, to get really started, you need funding. And I've already mentioned these interdisciplinary grant proposals, but also many opportunities for funding of clinical trials are available where you can get separate funding for translational endpoints. This is often the case. Specifically in the emerging field of personalized medicine and targeted therapies. And there it's usually easy to get money for the so-called ancillary studies or translational endpoints, where you look, for instance, at molecular biomarkers or gene expression-based biomarkers that predict response to the therapy. And then, of course, it's important that you get at least a working knowledge of the basic science that is involved and the methods that are involved. If you're working with a basic molecular biology lab that will do the single cell RNA sequencing for you, at least know how, in general terms, this kind of pipeline is working, and what are the sample requirements, how dedicated it is, what type of bioinformatics you use, what kind of expertise you need, what the cost is, and etc., etc. And you should then visit these labs and see how they work, and sit together with them and get at least some kind of understanding of what they are doing. Otherwise, it's very difficult to communicate with them on a level that allows you to have a fruitful and a successful collaboration. So biobanking is important, and it's the first step in this type of translational studies. And you can collect biological material, but of course you also need to collect the data. If you have just samples without annotation, that's not really very valuable. You need the clinical data. You need to know from what patient the samples were acquired, and also you need to know what treatments the patient got, what the outcome of the patient was, and the survival status, the recurrence status, etc., etc. And usually these data are gathered in a so-called LIMS, so a laboratory information management system that is linked to the biobank database, and that allows you to get a comprehensive overview of all the samples that were acquired in a patient, and the times, the dates that they were sampled, the status of the samples, etc., etc. In the former days, it used to be the case that all labs or all departments had their own biobank, and this was the case in our hospital. I remember that at one time we had 160 biobanks, so 160 different small units. Every one of us had one or two -80 freezers, and then everyone did his own type of labeling of separate limb system, etc., etc. But that's of course obsolete. You should not work like that any longer. You need to have one hospital-wide biobanking infrastructure that has a decent quality control, and there are ISO standards for that. You need to have a quality management system, standard operating procedures. You need to know what to do if something goes wrong. If, for instance, there is a power outage in the hospital and your freezer temperature starts to drop, and there needs to be an alarm, so that the technical department or the technician will be automatically informed and gets an alarm message so that they can come and rescue your samples. And that's just one small example, but of course there are many, many other reasons why you should have one encompassing biobanking infrastructure in the hospital. Of course, you cannot always just take samples in patients. It depends a little bit on what type of samples this concerns. If it is so-called residual material that is otherwise thrown away in the garbage bin, then it's, at least in Belgium, from the purely legal point of view, it's not required to give a specific informed consent. But in most cases, you need to have an informed consent. And in fact, if you want to do a specific clinical study based on the biobanked material, then you need a double consent. You need an informed consent, a broad informed consent. That's usually when the patient enters the hospital or gives a consent for an operation. She or he will sign a broad informed consent that allows you to biobank. And then you need to specify, or the patient needs to specify, what type of samples she or he agrees to be taken, blood, saliva, semen, etc., etc. He needs to know and to agree with the type of research that will be undertaken, so genetic investigations, molecular biology in general, but also commercial aims and commercial goals are enumerated on the informed consent, because you will be aware that the pharmaceutical industry has a large interest in getting these samples, and most hospitals do sometimes sell samples and or anonymized data to the pharmaceutical industry, and then the patient needs to know that her or his samples or data will be sold to the pharmaceutical industry. There's nothing wrong with that intrinsically or ethically, but the patient needs to give informed consent for that, of course. So that's a broad informed consent. But then if you want to do a specific clinical study using this material, then you need a second informed consent that is then applicable specifically for the clinical study or the translational study that you want to do, and not for the biobanking per se. So there are different types of biobanks. There are population study biobanks where healthy persons are being surveyed and where samples are taken to do epidemiology type of research. And there are basic research biobanks, translational study biobanks, etc., etc. And then clinical trial biobanks, of course, and that's the most common one, and the biobank that is filled quite rapidly. And all biobanks or all clinical trials rather have now usually translational endpoints where samples need to be stored. And then if it's an industry sponsored trial, of course, they are stored temporarily in the hospital or in the OR. We have freezers in the OR that makes it very easy to store samples. And so whenever I do a cancer surgery, I take a sample, of course, on condition that it's not interfering with, for instance, the margin status examination by the pathologist, but I take a sample and I ask the personnel, please put the sample in the freezer, and then the freezer is just a few meters away. And then every few months, this freezer, the contents of this temporary freezer is then emptied, and all the material is then transferred to the large biobanking facility, which is a few blocks away. The pathologists often have a biobank of their archived FFPE material. You know that in many countries there is a legal obligation to keep pathology material during a certain period of time. I know that in Belgium it's 30 years. So for 30 years you need to keep the blocks, the paraffin blocks, and usually a few slides are kept by the pathologists in a biobank. And nowadays you can do a lot on paraffin embedded material. You can do RNA isolation and transcription. You can do spatial transcriptomics on FFPE slides. So nowadays there's really a lot you can do on paraffin embedded material. And so it's not always necessary to freeze tissue or to extract immediately RNA or DNA. And there's a lot you can do with paraffin embedded material. And so this biobanking is, of course, not only applicable during the surgery, even though it's the most relevant step for us surgeons, but also at the moment of diagnosis. Biopsies are taken, core biopsies, fine needle biopsies, and also in the follow-up, patients can agree to provide material for additional biobanking. And maybe you have heard of these avatars. So avatars in cancer patients are based on samples that are taken in the patient and that are used to create either a PDX or a PDO, so a patient-derived organoid that is then used to probe chemosensitivity or sensitivity to targeted drugs or small molecule inhibitors, tyrosine kinase inhibitors, etc. in the animal, and based on the findings in the animal, the patient is then treated with drug A versus drug B versus drug C. So it's a very, very powerful, biobanking is a very powerful way to allow and to enable precision medicine in oncology in general. So just for your information, there are some international entities that are really busy with global biobanking initiatives, with creating international standards, with harmonization, with providing training, accreditation, etc. And this is one such example, ISBER, which is an international entity that is focused on biobanking. They have their annual meeting, as you can see here, in Seattle in 2023. And then there are national biobanking initiatives. I'm not, of course, very familiar with the rest of Europe, but in Belgium, at least, we have this BBMRI, which links, in fact, all the large hospitals, so both the academic, the university hospitals, and the large non-academic hospitals. And so this is, of course, a very powerful way. If you can expand the pool of samples, you really get thousands and tens of thousands of patients and samples. And this is really what makes you as a country attractive for the pharmaceutical industry and the drug discovery industry, to be an interesting partner. If you can provide samples, but also the data, so annotated samples. This is just a slide that shows you that it's because in the past we used to immediately extract RNA from the tissue samples and using this medium RNA later. But in fact, we now realize that this is not really required because the stability of the RNA is such that it really stays stable. And this is what is shown in these graphs. So the x-axis represents the time point, and so up to a few hours, like 3 hours. And then in the y-axis is a measure of stability. And you see in the left upper graph, for instance, and that in fact, the fresh frozen samples remain. So the quality of the RNA and the stability of the RNA is just as good in the fresh frozen samples compared to the samples where RNA later, this medium was used. So let's now go to some examples of opportunities for translational research. And of course, the organoids, the PDXs are already mentioned. And some of my colleagues who are doing sarcoma surgeries have now set up, together with the basic science labs and basic cancerology labs, they have now established a PDX sarcoma model. So both in bone sarcomas and in retroperitoneal sarcomas. Of course, technology is is being used on a daily basis, and there are examples of surgeons who are involved in developing novel robotic systems, novel hybrid systems, and like the IRCAT in Strasbourg is a nice example. And some surgeons have an engineering degree, and so this is of course a well-known field. Enhanced imaging is, I think, a very interesting field of translational research. I will give some examples. 3D bioprinting, tissue engineering, and then, of course, as already mentioned, the huge field and the booming field of artificial intelligence, or the so-called artificial intelligence. We all have seen the ChatGPT as another example. But of course, big data have quite some promise, although I must say that the real clinically relevant applications are not yet there. And it's not always easy to translate big data into important, how would I say, in practice changing algorithms or practice change that is relevant for patients. We've seen that in the COVID epidemic. Billions of vaccines, billions of patients, huge amounts of data, but the only real advances that came out of all these analyses using all these big data sets were the few randomized trials that were performed. And so I can see the potential advantage of machine learning and of big data, but I think it's really a big hype, and that we should be careful not to over-interpret the potential of big data. So this is an example of these organoids and tumoroids and PDXs. PDXs are, of course, much simpler, are much cheaper, because organoids are quite expensive, because these special growth media, you see that here in the middle of the figure, EGF, Noggin, etc. They are patented, as is often the case in biomedical research, and they are, as a consequence, quite expensive, these growth media. But in theory, it's of course an ideal model system for basic research, but also for patient treatment as an avatar. So to test chemosensitivity in patients where it's really difficult to predict what type of treatment will give the best response. And a typical example is a patient with ovarian cancer who has had a first, a second, and a third line treatment, and then what to do next, what to give next. And there are a number of options that you can test then using either a PDX or a PDO and see what happens in the mouse, and then use the same treatment in the patient where you got the tissue from. Then a few examples from image-guided surgery. So near-infrared, you know that, of course. It's using ICG, indocyanine green, for perfusion imaging, for lymphatic mapping, sentinel lymph node mapping, targeted imaging. But next to ICG, there are now targeted probes and more specific probes, labeled probes, that target CEA or other cancer-related epitopes. Hyperspectral imaging is now up and coming. We have our own project with that. So spectral imaging allows you not to image perfusion, but to image the oxygen level of the tissue. And it's based on the fact that saturated, so oxygenated hemoglobin has a different absorption spectrum compared to the non-oxygenated hemoglobin. You can exploit this difference to quantify in a semi-quantitative way, of course, the oxygen content of the tissue, and it then can generate oxygenation maps of the tissue, which is really quite nice. And then Cherenkov luminescence based on PET-CT for margin assessment, hybrid imaging, etc., etc. So this is from a semi-recent review a few years ago already, that gives you an overview of the targeted imaging moieties that are available now. All of them in preclinical settings. And so, as far as I know, none of them has yet has been approved for clinical use. And so they are targeting the VEGF receptor, integrins, etc., etc. So a very promising field. And of course, this is familiar for you. This is what we're going to do using spectral imaging. So we divide the visible spectrum into, well, and also the near infrared, so which is not visible to the human eye. We divide it in small blocks of, let's say, 5 to 10 nanometers, and then this is then used to generate semi-quantitative maps of tissue oxygenation. And this is a figure from a grant proposal that we are going to submit just now. We have already assembled a prototype of a hyperspectral imaging laparoscope, and we're going to validate it first on tissue phantoms, on blood samples, and then in large animals, and then as the last work package, we're going to do a clinical study where we will try to validate the technique and compare it with indocyanine green perfusion imaging in patients who will undergo surgery for esophageal cancer, where we will try to image the stomach substitute after resection of the cancer in the esophagus. And this is quite nice as a research topic. It's intraoperative margin assessment using this small cabinet, and the patient gets a PET tracer intravenously, I think in the morning or the evening before the operation. And then it goes into a cabinet, and what is detected by the apparatus is not the positrons or the gamma rays, as in the usual PET-CT scan, but the Cherenkov light. Cherenkov light is the kind of bluish light that you can also see in a nuclear reactor, and it's in fact elicited. You see it there when photons travel through the tissue at a velocity greater than the speed of light in that medium. This is an example from prostate cancer, where the margins are being assessed. And I know that in our hospital, they are using it for breast cancer. And so imaging of the cut margins of the breast cancer specimen. But I think it's quite a promising technique and a nice topic for further investigation. Tissue engineering and bioprinting, also a huge field, 3D printing, and of course, an excellent example. It's usually done by plastic surgeons, but it's an excellent example of bi-directional translational studies, where you can do all kinds of stuff, 3D bioprinting of cartilage, bone, and the future is probably the 3D printing of blood vessels, of complete organs, etc., etc. So artificial intelligence, a lot of applications, but in fact, the only field that is now a little bit more advanced is the annotation and classification of images, automatic feature recognition based on X-rays, on medical images. And all the other applications that are being hyped at the moment, yeah, we'll have to see. Time will tell. But at this point of time, I think it's mostly the imaging-based artificial intelligence and machine learning and deep learning algorithms that are really making a difference. And the classical example is the camera that is now being marketed for the automatic classification of skin lesions, where the algorithm will tell you the probability of the skin lesion being a melanoma or not a melanoma. And that's, I think, of course, a traditional successful example of the combination of machine learning and clinical medicine, which is relevant for patients. So drug delivery, many of the members of the ESO and also a complete working group is dedicated on drug delivery, intraperitoneal drug delivery, HIPEC, etc. And so I will give some examples of the translational research that we are doing. This is an example that is not from our department, but I think this is an interesting example. It is a way to apply chemotherapy directly to a cancer of the pancreas using electrically enhanced drug delivery, kind of electrophoresis, the same kind of electrophoresis that they are doing to separate DNA fragments in a southern blot. You can do the same to drive chemotherapy into the tissue, and this is already clinically used. It's in clinical trials where this device is implanted. It's put against the pancreas, on the surface of the pancreas tumor, and then this is attached to a battery using leads that go into the patient, a little bit like a pacemaker, and then a DC current is applied, and then the chemotherapy is driven through the tissue. And we have our own models to study aerosolized drug delivery. And as you may know, we have in vitro models, we have computational fluid dynamics models where we can model all kinds of parameters and the characteristics of the nebulizers, the shape of the electrical field. Here on the right side, it's electrostatic precipitation combined with aerosolized drug delivery. This is an example of a similar CFD model from another group. It's from a German group published in 2021. This is this is a video. I didn't realize that this was a video. But anyway, you can see here the 3D printed model that we are using. This here is a 3D printed model of a peritoneal cavity. It's based on a CT scan that we took in a patient with a pneumoperitoneum. In fact, it was a spontaneous pneumoperitoneum due to a perforation of a hollow organ. And then this was put into a CFD model and also 3D printed. And we are now using this artificial peritoneal cavity to perform additional studies like high-speed imaging of aerosol droplets. We are trying to measure the energy, the impaction energy of the droplets on the surface using piezoelectric materials. We are examining the interaction pattern of the droplets with the mesothelium, like sticking, splashing, fragmentation. There are a number of interaction types that are possible as soon as a droplet hits the mesothelium, etc., etc. So that's an example from our lab. Biophysics is one of the topics of our lab, and this is a device that we have assembled. It's based on Ussing diffusion chambers, but what we do here is a very different application. We use this to measure the hydraulic conductivity of cancer tissue, because this is one of the parameters that you need to measure if you want to make CFD models of the interstitial fluid pressure based on MR scans in patients. And that's now something that we are doing in a clinical study. That's another example of combination with physics. We are collaborating, in fact, with engineers and also with physicists in this field. It's electromotive drug administration, so it's not using an aerosol, but using a liquid, but where again we use electrical current, so a DC electrical current to drive the drug into the tissue and then see what parameters are relevant, like the power intensity, the voltage, the shape of the current, so either a block or a smooth, kind of a sine or a cosine wave, whether it's useful to reverse the polarity, etc., etc. So that's also a nice example of collaboration with other disciplines. So to finish, some final considerations. So I think that surgeons have all the assets to be successful in translational studies, because first of all, we have access to the tissue. And that makes us an attractive partner for basic scientists, but also for engineers, etc., etc. And also we have a helicopter view on the patient, on the disease, on cancer biology, and this is really an asset, because many of these basic scientists, they are very, very focused, and they spend their entire career on the characterization of one single pathway or one single molecule, like E-cadherin, to give one example of one of my colleagues here. But if you really have a helicopter view on what is possible and the potential clinical implications, and that's really, really an asset. And also, of course, many surgeons are inherently interested in technology and engineering, are very technically minded, and this helps, of course, to get sympathy and an understanding for what engineers are doing, and also to get quite rapidly an understanding of the underlying principles and the physics and the mathematics of it. It's really not that difficult. But what is challenging is, of course, to find partners in fields like physics or computer science who are not really sitting there waiting for you, just as the molecular biologists. And of course, but this is for research in general, you need to have protected time and enough energy to do it all, to combine busy clinical practice with attending congresses and supervising PhD students, and so time management and protecting your work-life balance is always a challenge, also in translational research and in studies. And then you need funding, but it is possible. You need, of course, to master the skills. You need to know how to write a decent English text, how to structure something, how to convince people using a written proposal, how to do statistical analysis, a sample size calculation, how to decently make figures, graphs, scientific graphs, etc., etc. But those are really the basic skills that you need to be successful in research in general. So on that note, I would like to end now, and I will now give the floor to Olivia, to the audience, and if there are any questions, I will be very, very happy to try to answer them. Thank you.
Thank you very, very much, Wim, for this very interesting journey through every single aspect of translational research that could involve surgeons. And I'm sure there will be a lot of questions, or at least I hope so, because I took a glance to the auditorium tonight, and I saw that we have really different levels. We have junior surgeons, but we also have a lot of senior surgeons. So I'm sure that they will have questions and from different perspectives. I was wondering, because you're also in the board of ESO, and can you tell us anything about how opportunities are present depending on different countries for junior surgeons in the field of research, of translational research?
Well, that's a very good question. It's usually based on so-called ad hoc opportunities. But it depends a little bit on the specific field. I know, for instance, that for clinical studies and for clinical research skills, there are initiatives by the ESMO and also by the ESO, but specifically by ESMO. They organize research seminars that allow you to get basic skills in clinical research. And then what they do is they invite you to bring your own protocol, and then this is discussed by statisticians and methodologists. And this really helps you in developing your skills in clinical studies. But for translational research, it's very much depending on the specific topic. But what you should do is contact other centers where you know that the expertise is available, and either yourself or your chief of the department of someone who you know is really well acquainted with the other team, and just ask for an opportunity for a research visit. but it's usually very much on an ad hoc basis. I'm not aware of any formal ESO or ESMO based training courses for translational research, because of course it's a very, very broad topic, and it's very different if you want to do an artificial intelligence or machine learning based project compared to an omics based project, for instance. And so that's the reason why it really depends on, I think, the specific expertise that you need.
Thank you very much. We have a question from the audience, from Carmela. She asks about, in terms of analyzing results from translational research, what additional statistics courses should surgeons learn about?
Well, if it's omics, then that's not something you can do yourself, because then you need a bioinformatician. This takes a master degree to be able to analyze, for instance, the output of a single cell sequencing experiment. It's gigabytes of data, and you need very specific tools. But I would say for basic biology experiments, the statistics are usually much simpler than the statistics that you need for clinical studies. And for instance, sample size calculations are usually not performed. If you do mouse experiments, it's 2 times 5 mice or 10 mice. And although it's often required by funding agencies, usually sample size calculations are not performed for animal studies, and the statistics are quite simple. You can use a GraphPad, which is commonly used, or any one of these quite simple statistics programs. So I would say that for basic animal research, the statistics are usually easier rather than harder compared to clinical studies. I think the exception is bioinformatics, where you really need a bioinformatician to help you. And you must, of course, make sure that you have an understanding, a basic understanding of what the output looks like, what a heat plot is, and what dimensionality reduction is, etc., etc. But that takes you like a few hours to read something about it and to get a high level understanding so that you can communicate and that you can help in drafting the legends to the figures. But to do the bioinformatics analysis needs a specialist, unless you have your own master degree in that field. But that's usually not the case, I think.
Thank you. I found it very interesting that many of the translational fields you were talking about in your presentations required some sort of engineering. And you also said at the end of the presentation that finding the right partner is one of the important keys finally to have good translational research. How do you proceed? Do you get inspiration from the potential partners you meet in different fields, or do you look for what you need for a project you imagined?
Yeah, well, it depends. If I really have no idea where I can find a specific expertise, I go to PubMed and I type in the technique or the expertise that I need, and also the location, Ghent University. And this may sound silly, but I'm sure that many of you have that experience, that you are not aware of what your neighbor is doing. I mean, if you work in a large university, sometimes you realize that the person who is in another building, the other side of the city, is doing something that is really relevant for you and interesting for you. And so often this is the way that I find potential collaborations, or for a machine. I was looking for a tool to, I don't remember the specific technique, but I wanted a specific technical device, and I did it the same way: PubMed and then location, Ghent University. And if it's not available in Ghent, I looked at PubMed Belgium to see where else in Belgium it's available. But that's of course the last resort. Usually you are more or less aware. For instance, in immunology, I didn't talk about immunology, but that's also a field we are now quite active in. And of course, once you have a certain seniority in research. You get asked as a member of panels, ground review panels, and then you get to read a lot of reviews and ground proposals, and then you know who is busy with cancer immunology in your center in Belgium, internationally, and you get to know these people. But often you don't really know what your neighbor is doing, and then you need to depend on search engines or on initiatives that are often now organized, at least in our center, to bring together clinicians and basic scientists. And I think that's really very, very useful. If universities really invest in joint meetings or in social events, like science cafes, stuff like that, that really bring together clinicians, but clinician scientists, so physician scientists, surgeon scientists, and basic scientists, either informally or formally, just to get to know each other and to make people realize, oh, this can be interesting for me, and she or he can be an interesting partner. Yeah, that's usually how it works.
We have two connected questions. One is from Amit Gupta from India, and the other one from Sigurd Folkvord. I think he's from Brazil. Amit is asking if the residents could be sent, I think, from India when they are in surgical oncology to on an exchange programs for research projects abroad, and I think, notably in Europe. And Sigurd wants to know what would be a good starting point, either a fellowship or a course for surgeons coming from countries where research and grants are difficult to acquire.
Yeah. Well, for the first question, it depends on what you want to do in the country or in the center that you are visiting. If you really want to participate in clinical practice and in surgeries, doing outpatient clinics, participating in surgeries, then of course there's the language, and then there's the legal requirements. Every country has its own specific requirements to allow you to practice medicine. So that's of course a major issue. But if you don't want to be involved in clinical practice, then it's really very easy. And then you just write to the head of the lab, please, can I go for a visit, either a funded visit or a non-funded visit, and many labs offer that opportunity, just to visit, to learn a technique, whatever. So international mobility is really very, very high in the field of basic research. It's very, very common to go and visit another center. Even senior colleagues who I have, they regularly go for a sabbatical where they stay several months in a well-known US or wherever university to learn a technique, to set up collaborations. So this is very common, and it's quite easy. Of course, to get a funded position in another center, then it depends on the availability of funded PhD positions, and then you need to look on the website or ask, please, do you have any funded opportunities? And then of course it's based on a competition. Then you need to submit as a candidate, and then there will be a range of other candidates, and then there will be an interview and a selection, and that's how it works for a funded PhD position. And then for the question about funding, not all research is expensive. I mean, biological research is usually the most expensive because you need animals, you need antibodies, you need cell lines, you need a lot of techniques, a lot of machines. Sequencing is expensive, but you can do very relevant research that is not expensive. You can do outcomes research. You can do research that is focusing on quality of life, on improvement of whatever method or implementation of healthcare. Also, all the research that is based on artificial intelligence, machine learning, stuff like that, it's cheap. Python is free. R software, which I'm starting to learn now. It's free. Server time, storage space on Google, it's on Amazon. It's not free, but it's very, very cheap. So a lot of research that you can do, you need the skills, but you don't need a lot of money. So I would advise you, if you want to get involved in machine learning, there are excellent online courses. Learn how to code in Python. Learn how to set up an artificial intelligence algorithm, and you don't need to be an expert in mathematics. It's really quite easy for someone who has a basic knowledge of, let's say, mathematics and how programming to get successful and to be successful in this field. So it's not always expensive. And then, of course, the key to success is collaboration, as already mentioned a few times. Collaborate with colleagues in other fields, but also collaborate with other surgeons. That's why some countries like the Netherlands are so successful, because they collaborate. In France, you have the Prodige, you have the RENAP. In Italy, and you now have large networks. In the UK, you have the Surgical Trainees Network. So if you start collaborating and forming networks within your country or even between countries, I think that's really a key to success.
Thank you very much. And I think I have two more questions. So for a senior surgeon this time, what do you think it would be who wants to perform research in surgical oncology? What do you think would be that optimal maybe time balance between translational activity and surgical activity?
Yeah. Well, for me personally, I work half-time, 2.5 days clinical, 2.5 days purely academic, so research and teaching. I know this is rather the exception than the rule. It's not always easy, but if you work in a university hospital, which is not based on if you're in a salaried position, and everyone is in a salaried position, then it's easier. Because if you're in a group, in a department with, let's say, 10 staff members, then it's usually the case that some of these staff members don't really have an interest in research. They like to be clinically active, they like to teach residents and to teach medical students, but they don't feel the need to do research. Whereas others are really interested and devoted to research, and then you can come to an agreement that some people have more protected time than others. But then, of course, in exchange, you should also help your colleagues who are less involved in research if they want to publish a paper or do some analysis, that you help them. But that's how we do it. So we have a number, of course, the work needs to be done, and we all are busy in clinical practice, and patients need to get treated. But that's the best way. Make sure that you are in a department where you have a lot of staff members, and that you get an agreement. I can do some more research, have some more protected time, but in exchange, I will do this and that for the others to help them. But I would say that if you want, you need at least 1 day per week, 1 in 5 of protected time. Otherwise, it's quite impossible. And I would say 2 is, in fact, 2 days is, I would say, ideal. But 1 day is the minimum. And what you do not want to do is to spend every evening and every weekend to write papers and to write grant proposals, because you will, you can do that for a few years, but if you do that for 10 years, you will, the risk of a burnout is really quite high then. And so you should always guard your work-life balance. Yeah.
So the last question, I think, before wrapping it up, comes from Carmela, and she would like, it's a reformatory question, because she would like to know what additional subjects or topics should be included in the surgical oncology curriculum in order for a surgeon to be ready for translational research.
Well, it's been some time since I've seen the curriculum as it was published in the European Journal of Surgical Oncology, but it's already quite encompassing. It includes everything from clinical trial design to basic science, etc., etc. But yeah, I think it depends really a lot on the field that you want to get involved in. So as already stated, if you're interested in machine learning, please start working with Python. It's free. The courses are free, and there's a huge online community. A lot of surgeons are involved with it, so you can learn from your colleagues. And then for more biology and biomarkers and omics, probably a basic course would be useful in how these techniques work. What is a PCR? How does a PCR work? What is a southern blot? How does Sanger sequencing work? How does NGS, so next-gen sequencing work? What are the restrictions? What can you expect from the output? What are realistic expectations, etc., etc. So maybe that's an interesting additional field that is not yet covered, I think, in the SO curriculum. So a basic science, a basic course in omics, tools and technologies, and a basic understanding of the bioinformatics pipelines and platforms that are being used. Yeah, so that would be a first suggestion, but yeah, I'm sure there will be other proposals and additional proposals that are possible.
Thank you very much, Wim. It was an incredible helicopter flight over the field of translational research. I'm sure that's going to be very crowded on Python tonight with everybody trying to get some machine learning courses and coding. Thanks again to our audience for the questions, and thank you for Maria for hosting on behalf of ESO. And I'm sure there's going to be more webinars coming soon from our side.
Thank you, everyone. Okay. You can have a look also on our website for the upcoming webinars for February in the courses. And we'll be seeing you again soon.
Thank you.
Good
Thank
night.
you.
Have a nice evening.
Have a nice evening.
Bye-bye.
Bye.